Good evening, everyone. We'll just wait for 4 to 5 minutes if anyone else is joining, or else we'll start with the session. I hope the screen is visible to all of you. I mean, the people who are there in the meeting. I would request all of you to kindly mute yourselves so that there's no disturbance while we proceed with the class. If at any point during the session any of you feel like asking anything related to the topics that we are discussing, please feel free to write that in the chat box so that I can address your queries during the session itself. Just one more minute, we'll wait so that all those who are joining, we can let them in the meeting room and then we can like smoothly start the session. Oof, so- Okay, so I think we can start with the session now. So if anyone else would join, I'll let them in again.
పిల్లలు ఉంది.
Okay. Okay, so I hope my screen is visible to everyone and I'm also audible to everyone. If at any point you guys feel like, there's lag in video or just a second. Okay. Okay. So this is your week one NPTEL live session. So as you all know, you guys have enrolled in experimental nanobiotechnology, which is a 4 week course basically. So you guys already have the course content, what would be the syllabus and the pre-recorded sessions by the instructor, Professor Gopinath. So in this session, we'll mainly be focusing on whatever topics are already being covered there and whatever like there are pre-recorded videos. So you guys would have watched those videos for week one that are available on NPTEL website. So after that, we'll just be quickly revising all those topics, some of the basics of those topics, and in addition to that, what we'll be doing, we'll be solving some of the previous year assignment questions as well. So, in this session, also we'll follow the same pattern. We'll start with, whatever the course content is there. Just a second, okay. So,
हेलो
yes,
गुड इवनिंग मैम
good
मैम
evening.
आई हैव वन प्रॉब्लम मैम मैंने एनपीटीएल कोर्स ज्वाइन तो कर लिया है पर मैम मेरा जो अनाउंसमेंट ग्रुप है उसमें मुझे ज्वाइन नहीं किया तो मेरे मेल पे कोई वो नहीं आता है ये जो मैंने क्लास ली है वो मेरी फ्रेंड के मेल से मैंने अपने उस पे मंगवाई थी तब मैं ज्वाइन कर पा रही हूँ तो मैंने मेल भी बहुत बार किया है पर कोई रिस्पॉन्स ही नहीं आ रहा उधर से मैम
I think you can write your name maybe in the chat box and your email ID as well. I can maybe like share to my NPTEL coordinator and maybe they can help. Or else, basically, it would be like you can reach out to the helpline number. There is one calling number as well that you can find on Google. So in addition to that, I guess mail is the best option. So in that announcement group, but still, like if you have the I mean the Google Meet link, you can join in. The sessions will be having Friday, Saturday, and
यस
I
मैम
think Monday, 6 to 8 p.m. There would be live sessions so that you can
यस मैम
join. So otherwise, usually they resolve these issues over mail only. But if in case it's not possible or it's getting delayed, so I have
यस मैम
informed like you can do that. I mean, join the session Friday, Saturday,
मैं
6
अपना
to 8
नाम
p.m.
और जीमेल इस चैट बॉक्स पे भेज दूँ?
Yes, you can do that. I'll
ओके मैम,
take a picture of that and maybe I can forward that.
ओके मैम, थैंक यू मैम।
Okay, all right. So let us start with the session now. Uh, but before that, let me introduce myself. My name is Anusha Srivastava. I'm currently doing teaching assistantship under NPTEL. In addition to that, yes, you will get the recorded version of this class as well. But once the class is finished, and that too on the YouTube link, you will be getting a YouTube link in that would have been shared with you. Along with the YouTube link, you will also get access to this presentation that I am discussing right now for all the weeks. So you'll also be getting the recorded sessions on YouTube, whatever we are like this live session, And in addition to that, you'll also be getting the this presentation that we'll be discussing right now. Okay, so one request to everyone, uh, if everybody would join on time, it would be smooth for me to conduct the class instead of coming back again and letting everyone in. Okay, so for this week's session, the course content, we'll basically be talking about what are the different, some of the very popular mechanisms for synthesis of nanomaterials, and some of those techniques. In addition to that, we'll also be studying about the synthesis of different inorganic nanomaterials, sorry, nanoparticles, and biological synthesis. How do we perform the biological synthesis? Then synthesis of some, carbon dots, and synthesis of polymeric nanoparticles. So, if some of you are not aware, one thing I would like to inform that there is one course, you know, uh, biomedical nanotechnology, that I guess runs in the this July to December session. So that is also a basics to, like, right now we're discussing about the experimental aspects of nanobiotechnology, like what can be done, what are those the applications of the nanoparticles and all those things. But there's also a basic course that you can, look for biomedical nanotechnology that runs in the different semester. So let's start with it. So, whenever we are talking about nanoparticles, so there could be different approaches for its synthesis. Suppose there is one top-down approach and the other one is bottom-up approach. Now, before diving deep into it, let us first understand that whenever we are talking about nanoparticles or nanomaterials, so anything that has dimensions in the nano range, like nanometers, like below like 1,000 nanometers, we consider it as a nanomaterial or nanoparticles. So for the top-down approach, what is happening is you're taking a bulk material and then coming down to nanoparticles. Okay. So suppose you can visualize it. Suppose you have a wall that has been made of bricks, different bricks. Now, this is the top-down approach. Suppose there are different bricks. Now you want to obtain single bricks. You kind of break it, and then you have these single bricks. Okay. Now, and this one is the top down approach. Now the bottom approach could be like you have cement, you have all those cement, and then you synthesize, or I should say you can make these individual bricks. Or suppose you are coming down, suppose you already you have these bricks and now you have to make a wall. So that's kind of like a reverse kind of thing. In top down approach, you have a bulk material and then you come down to nanoparticles. So this is the end point. In bottom approach, what is happening? You have smaller particles and then there is nucleation that follows, and after nucleation, you finally get your nanoparticles. These are the two, I would say, approaches for synthesis of nanomaterials. Just a second. Okay. Okay, moving forward. So this synthesis of nanomaterials, it can follow different routes as well. You can do physical, by physical means, hybrid means, biological means, and chemical means as well. So there are different methods for synthesis of nanoparticles as well, whether you're following a physical method, a biological method, or a chemical method. Now there are differences in that as well. So whenever we're talking about physical method, you are kind of, you use ultrasonication, laser ablation, irradiation, evaporation, condensation. Like we'll be discussing all of these in details, like how this happens and what exactly is the mechanism for this. Biological means most of the time whenever we are talking about biological method, it refers to green synthesis. Whenever we are talking about green synthesis, we'll also look deep into it, like what does exactly green synthesis means. like free of organic solvents, free of salts. So in these cases, we basically use plants and microorganisms. Like you guys would have also heard about like these plant extracts are being used for synthesis of different nanomaterials or nanoparticles. And apart from that, a lot of microorganisms are also currently being used for, for as cargo for drug offloading. Suppose the virus-like particles that are currently available or viral nanoparticles. So in that case, the infectious material from the virus is removed and whatever is left behind, this cargo, this is particularly used as a nanoparticle in which you load different drugs and then which can be used for different nanoparticle based applications. So we'll be looking all of this in a little detail. So first thing that was there in your, the pre-recorded session as well is the mechanical milling, this is, that we also call as ball mining technique. Okay. So what happens in this? Suppose, okay, so anyone has any doubt? Somebody raised hand. You have any doubt?
Yes, ma'am. Good evening. So I raised a hand and I've seen the lecture regarding ball milling.
Okay.
Um, I have a doubt in regard to the characteristics of ball milling, I think, which you will explain later in the slide. So do I wait until you come till there?
Sure, sure. We'll
No,
be discussing
thank you.
about all those things and some of the features as well, like what could be the approach, what could be the parameters, and how can we optimize, how can we get the monodispersed kind of nanoparticles through ball milling. Although we'll also be discussing some of the questions, and in addition to that, what are the shortcomings of you know this technique. So in traditional approach that we are talking about right now, mechanical milling, which is high energy ball mining, is a top down technique. Just now we discussed like we have the bulk material and then we come down to nanoparticles that we are synthesized. So this is the top down approach. So ball mining or mechanical milling that we is a kind of top down approach where bulk materials are repeatedly fractured, cold welded and re-welded inside a rotating mill to produce nanoparticles. Now, what is the milling media? Here, these small balls is referred to as the milling media, which are made of ceramics or zirconia or these kind of materials, and these are the I would say the bulk materials that are there. So the pressure or force that they exert, that these milling media or these balls exert on these bulk materials, because of that, these nanoparticles are formed. So to produce nanoparticles, the process uses hardened balls in a mill, example planetary or attritor mill, to generate high impact and shear forces that reduce particle size to the nanometer range. Similarly, so there is bulk material. So because of this shear forces and high impact that are being exerted by these milling balls, these mining balls, uh, results in particle size in nanometer range. So this is kind of a cost-effective, scalable, and suitable for metals, alloys, ceramics, and composites, though contamination and agglomeration can occur. Now, so this is one of the technique by which these nanomaterials are formed. Now, let us understand a little deeper into it. So one problem that any of the techniques, whether we are talking about this ball milling, whether we'll talk about self-assembly, whether you will talk about synthesis of nanoparticles via ultrasonic atomization, all of these techniques, like if you're not taking proper care of it, if there is not proper capping or shielding, there would be aggregation of nanoparticles. Now, what does aggregation actually means? Suppose these are your nanoparticles, okay? So aggregation means if they are not properly, there is no proper force of repulsion, they would come together and kind of aggregate. And once they aggregate, these aggregates are formed, they lose their inner characteristics. So there are different methods by which this aggregation can be reduced. Now that we'll also discuss, but before that we'll study these nanoparticles. Why does these nanoparticles aggregate? So they aggregate mainly due to the following reasons. First reason is high surface energy. Now, as we know that suppose this is the bulk material and this is small size nanoparticles. So obviously for this one, the the surface to volume ratio would be high for the nanoparticles. So because of this, because of high surface to volume ratio, it makes them thermodynamically unstable. So they stick together to reduce total surface energy. So we all know that every system, proceeds towards from high energy to low energy state in order to attain stability. So these small size nanoparticles which have a large surface to volume ratio, they have very high surface energy. And in order to reduce this very high surface energy, they tend to aggregate, they tend to agglomerate. So next reason is strong interparticle forces. So the van der Waals forces that kind of operate between these nanoparticles and the magnetic forces, the electrostatic interaction, all of these also come into the picture. They also have a nice interplay because of which all these nanoparticles come together and kind of aggregate. Then insufficient stabilization, lack of proper surfactant capping agents. Now there are different kind of capping agents available. Suppose this is your nanoparticles. Then what you do, a lot of times we use capping agents to cover these nanoparticles. That is why they are called as capping agents, because of which the they would not come closer to each other and there would be no aggregation. So capping agents are a must, or surface charge low zeta potential. We'll discuss about when we'll study about the characterization of these nanoparticles. What are the different techniques that are used for characterizing the nanoparticles? We'll study all of it, like how do we characterize nanoparticles, zeta potential values, how do we measure that, dynamic light scattering, all those things we'll see. Now, okay. So, or surface charge allows particle to collide and permanently attach. So, in this also, as you can see, this is the bulk material. Okay. Then after mechanical grinding, if you're not adding surfactant, if there is no capping, particle aggregates due to high surface energy generated on the milled particles. But if you add surfactant, suppose this is your nanoparticles that are formed, and then you suppose this is positive in nature, then you are adding anionic surfactant. Now it will surround the nanoparticle, and then there would be repulsive forces operating between these, and there would be no aggregation. Same if you are having, anionic bulk material, then you are adding cationic surfactant, and in case of neutral you are adding non-ionic surfactant. So these are the methods. Surfactant molecules, what does surfactant molecules do? They get absorbed on the surface of the generated particles. Suppose these are the generated particles, they get absorbed on the surface of generated particles and helps in lowering their surface energy by creating an organic layer on them and thus preventing them from aggregation. Now this aggregation issue, just a second. Okay. I'm really sorry for the disturbance, but I have to let every student in. Okay, let's come back to the point. So we see how ball milling operates, how nanoparticles are produced, and if we are not considering this problem of aggregation, or whatever nanoparticles we are forming, they would come together, they would aggregate because they have very large surface to volume ratio, and because of that they have very high surface energy as well, which makes them come together and aggregate. So in order to prevent them from aggregation, what we do we add surfactants, we do capping so that they do not come together and aggregate. Moving forward, now whatever, like in these two slides that we have studied, and whatever you would have watched in the in your pre-recorded session, just try attempting this question. Just try attempting this question. I'll just wait, like give you like kind of 30 seconds or 60 seconds, and you guys can try and write your answers in the chat box, and then we'll discuss this. So these questions are from your previous year assignments that were uploaded. Okay, just 15 seconds more. Okay, so just a second. So obviously the correct answer is smaller media. Now why smaller media? Just a second. Okay. So just a humble request to all of you. It would be really great if all of you can join sharp at 6 p.m. so that I do not have to go back and let people in after that. Okay. So why smaller media? So whenever we are talking about this ball milling technique, now what you see here, these are different sized balls, and this is called as milling media. Okay, so why smaller media? Because smaller media means more particles per unit volume. Suppose if we are talking like this is the, you know, the mill in which these are media, and then there are your these nanoparticles. Now, if these these balls would be small, so smaller media means more particles per unit volume. Okay, inside the mill. So more media means more collisions. If they would be small in size, like these are large. Suppose for the same in the same mill, if there are smaller milling media or smaller balls. Remember, whenever I'm saying media, media means these balls. Milling media. Milling media are these balls. So what they would do, there would be more collisions, and because of more collisions, there would be faster and more uniform grinding. Because what we are doing ultimately, we are kind of grinding it. Whatever bulk material we have, with the help of these balls, we are mechanically grinding them so that they come to a small, or I would say, like precisely say in the nanometers range. So this is specially beneficial for fine and ultra fine grinding. So in order to ensure that whatever nanoparticles you are producing are fine, and that undergoes ultra fine grinding, it's better to choose smaller media. Now, what are some of the shortcomings of this mechanical milling technique? One of it is it produces broad particle size distribution, poor uniformity. Now, let us understand what does this particle size distribution mean. Whenever we are talking about nanoparticles through any technique, okay. Now, some of the techniques are very precise. Now, we'll study about this synthesis of nanoparticles by ultrasonic atomizer. Now, such kind of techniques ensures reproducible and monodispersed particles. Otherwise, all of these techniques like self-assembly, or maybe for that matter, desolvation also, if proper capping protocols are not followed. So it's not always like all nanoparticles that are being produced are of same size. So when all the nanoparticles produced through any synthesis technique are of same size, we call this as monodisperse. Okay, but in case whenever suppose you are following some technique which are yielding nanoparticles of different sizes. Okay, now in that case, this is called as polydisperse. Polydisperse. Okay, so this particle size distribution, what does it show? Whenever the nanoparticles that are formed are polydispersed, it would have a long particle size distribution. Like suppose your particles that are being formed, it can range from 50 nanometer to somewhere around suppose 600 nanometers, somewhere around 600 nanometers. So that is polydispersed particle with broad particle size distribution. But in case of monodispersed particle, if your particles are monodispersed it's mostly of same size, then what would happen? You will have a very narrow particle size distribution. Suppose this kind of spectra you will get. Now this is basically recorded when you are recording the DLS, dynamic light scattering measurement you do, and you record that. We'll study that. But for right now, just you understand what does mean by particle size distribution. Okay, so this means your particle can range somewhere from 10 to 100 nanometer or 10 to 50 nanometer. So this is like narrow particle size distribution, and this one is considered good. This one is considered ideal. So mechanical milling, one of the, just a second. Okay, now coming back. Okay, so the next shortcoming of mechanical milling is can introduce contamination from milling media. Okay, so this one is another shortcoming because these milling media, whatever particles they are being made of, like they could be made of zirconia, they could be made of ceramics, whatever particles they are being made of. So while in the process they are being grinded, okay, so there is definitely a huge possibility of contamination from milling media. So your nanoparticles, your bulk material that you are grinding, it could get contaminated from the material with which these milling media are made up of. Then high energy consumption. It consumes a lot of energy because this is mechanical milling, obviously. Then it may cause structural defects or or amorphization in the materials, difficult to scale for very fine nanoparticles. Like for if you want to synthesize less than 50 nanometer nanoparticles, it is very difficult to scale it. Difficult to scale it means in order to produce more amount of nanoparticles of less than 50 nanometers, it is difficult to achieve this through mechanical milling. So these are some of the shortcomings of mechanical milling and the basics that we already saw. Now try attempting this one. Okay. So I would request you to try giving answer for this one as well. I'm really sorry. Mhm. Okay. Okay, just 10 more seconds for rest of the people who have not yet done. Okay. Okay. So the correct answer is both A and R are true, and R is the correct explanation of A. So this one, so it says the vial in ball milling is typically filled to one half or one third of its capacity. So whatever this ball in ball milling setup is typically filled to one half and one third of its capacity. The reason is this empty space allows milling balls to move freely and generate stronger impacts, which enhances the attrition process. So obviously both of these points are correct because if there wouldn't be much space these milling balls won't be able to move freely and generate the kind of impact that is necessary for this, the formation of these nanoparticles. Now, the another technique is this and etching or chemical drying. I think there is something. Okay, so anyone wants to ask something?
Yes, ma'am. I have one doubt. So I had seen sir's video for preparing the ball milling at the end of his the video that he has showed. In that, he is using balls of both sizes. He's using a 5 ball and 2 ball. So
Mhm.
if 2 lesser the size gives you finer particles and quicker speed, then why use both? Why mix two balls? It's just for cost efficiency, or
I think in that video, you suppose, see, if in simple words I would try to explain, we were in that video, it was more of like a kind of optimizing that which balls would be best, and then sometimes, like for example, in a simple example, suppose there is one drug in most of the papers like that are coming. Suppose if we are talking about cancer, there is one drug that is doing the purpose, that is serving the purpose and doing it best. Now, what we are doing currently, the approach is we are kind of combining two drugs and using to target same cancer. So then there would be kind of synergistic kind of effect, because of which both of the drugs would be, contributing as an anti-cancer compound. So in same way, in that video, in that pre-recorded video in which sir has demonstrated, like he has used both kind of balls. So one purpose is like kind of optimization, that which one is performing better if it is, and maybe in some instances for some particles, if we are using balls of two different sizes, when combined together, it could also lead to more ultra fine, or uh, the grinding mechanism would be better, and there would be more impact or forces because of which nanoparticles are being produced. So this could be one of the reasons.
understood, ma'am. Thank you.
Okay. So moving forward, like this nanoparticle synthesis by etching. Now, etching is another method. Like first we understood ball milling and what are the shortcomings of ball milling and what are the different parameters that we need to take care of in order to achieve monodispersed kind of nanoparticles. Why they aggregate. Just a second, I think I can join from my phone also, so that if anybody is joining in, I can let them in directly from here. Okay, okay. So let us understand what exactly means by etching. That is chemical, and now etching are of two types. One is chemical etching and the other one is dry etching. This is also one of the methods of preparing nanoparticles. So nanoparticle synthesis by etching is a top-down approach. Again, just like mechanical milling, this is a top-down approach. Okay. This is a top-down approach. I hope my voice is not echoing. If in case it's echoing, please let me know. So the first one is chemical or wet etching. So in these cases, we use acids, bases, or oxidizing agents selectively, which dissolve parts of material, to release or form nanoparticles. Control of concentration, temperature, and time determines its final size. Now, one thing, whenever we're talking about nanoparticle synthesis, about synthesizing particular kind of nanoparticles and tuning its properties, its size. Because see, there could be different size ranges of nanoparticles. When we'll study about gold nanoparticles, we'll see how effectively, how beautifully the size can be tuned. Now, when we're talking about chemical etching, so this is, as you can see, this is copper stabilizer, silver stabilizer, then buffer, Hassel toy, silver stabilizer, copper stabilizer. In addition to this is FeCl3 or alcohol solution. So, based on that, and this is like there are different kinds of etching. One thing is the chemical etching, in which we are using different chemicals for finally synthesizing this nanoparticles, and the other one is dry or plasma etching. So reactive gases or plasma remove material layer by layer. So here the material is, suppose this is the sample, it would remove the material under controlled condition, enabling precise nanoscale particle formation. There would be nanoscale particle formation. Key advantage, good size tuning and surface modification. Limitations, obviously, if we are not taking care of the aggregation, there would be possible surface defects in aggregation and as well as chemical contamination. Other one is laser ablation method of nanomaterial synthesis. So in this case, what is happening? Suppose this is the fiber laser which is being used as a source of causing nanoparticles to be uh, sputtered out. So okay, so these are the nanoparticles. This is picture target. So whatever laser beam is there after passing through the mirror and this lens, it is getting concentrated on this target material. And based on that, when this laser falls on this, so what happens? Laser ablation is a versatile method for synthesizing nanomaterials involving the vaporization of a target material. So suppose this is a target material. This could be of any, maybe you can consider of something metal alloy, or you're forming copper or silver or gold nanoparticles. Target material with a laser to create a plasma plume. So there is creation of this plasma plume which then condenses into nanoparticles. Now there would be plasma plume which would finally condense and we'll get these nanoparticles. So in this case, what is the driving force? This laser. This laser which is falling on this target material and causing vaporization of this material. And once this material vaporizes and then when it condenses, it forms these nanoparticles. This technique is valuable for producing high purity nanomaterials including semiconductor quantum dots, carbon nanotubes, and core shell nanoparticles without the need for chemical precursors. So one important thing that you need to focus on for this particular method is there is no chemical precursor involved, because this is the target material, this is the driving force, this laser beam that is falling on it. So there are no chemical precursors involved. So this particular kind of method, because there are no chemical precursors or harsh chemicals, organic solvents being involved, we label it as green synthesis. And usually, uh, in today's time, these green synthesis is mostly preferred. So same thing, this target material is there. When laser beam falls on it, there is plasma plume that is created. Okay, so there would be evaporation, vapor formation, plasma formation, and then these clusters would be formed. Nucleation, what does nucleation mean? These smaller nanoparticles will keep on increasing in size and form primary particles. So these would be the nanoparticles. And if we do not address the aggregation over the time, if it is not there's no proper surfactant, if it not there's no any means followed for its to prevent its aggregation, it will eventually aggregate. Now, other method is sputtering. What does sputtering means? So in this case, what happens is, suppose this is the target material, these are magnets put above. This is target material. Suppose this is made up of chromium. So sputtering is a physical vapor deposition technique used to synthesize nanomaterials by ejecting target material atoms using energetic ions. Now, in the previous case, laser was being used to eject out the nanoparticles. In this case, there are energetic ions, and then it is being deposited them as thin films on a substrate. Okay, it is a versatile method for producing nanomaterials with control thickness size and structure, particularly for thin film applications. Now, this is the substrate heating, this is heating stage. Okay, here is the substrate that is being kept, and then this formation of thin film with physical deposition of sputtered particles. Suppose this is the substrate or the thin film on which we want a thin film layer of particles to be deposited on this particular substrate. So from this side there is argon sputter gas flowing in, because here it is written energetic ions. So for this purpose, this argon sputter gas is used, which is flowing, and then uh, this is the this is from this point it is going out. So ejection of sputtered particles with AR plus bombardment. So it is kind of bombarding on this, and then there is these particles are uh, this particular substrate is getting sputter coated based on the chromium that is falling down. So this is also one of the techniques for synthesis of nanoparticles. Other one is electroexplosion of wire, EEW method. So this electroexplosion of wire method is a technique used to synthesize nanomaterials by rapidly vaporizing a thin metal wire with a high current pulse. So in above two cases, there were a target substance. So in one case, it was being vaporized, and then when it was condensing, the nanoparticles were formed. In the other case, the high energetic ions were being used to bombard on the target materials, and then because of the sputtering mechanism, the nanoparticles were being formed. In this case, what's it there? This one is copper wire. So there is electrode in there. So this vaporization followed by rapid cooling leads to formation of nanoparticles. So more or less the mechanism is same, and all these methods are considered as green synthesis because there are no harsh chemicals that are being involved. So EW is a versatile method that can produce various nanomaterials including metals, oxides and nitrides by controlling the ambient medium surrounding the wire. Now, coming to the next topic, which is synthesis of gold nanoparticles. So, gold nanoparticles are one of the most used in biomedical application for a variety of purposes. So, we'll also look for its application, like what are the applications of these gold nanoparticles. Just a second, let people know. Okay. So, synthesis of gold nanoparticles. So, there are different mechanisms through which we can synthesize gold nanoparticles. One is the chemical reduction method. Now, these are very classic, I would say, classical synthesis methods. That is why this one was named as Turkish method. So, in this case, the precursor becomes this aurichloride. Gold salt such as HAuCl4 is reduced using trisodium citrate. So this one is used as a reducing agent, and this is the precursor in boiling water. Citrate act both as reducing and stabilizing agent. Now you have to focus on this one. Like this trisodium citrate is not only helping reduce this precursor in forming gold nanoparticles, but this citrate also act as a reducing and stabilizing agent. Like it will form a layer around it, because of which it would be like it would stabilize, producing spherical gold nanoparticles. Now again, if you see here, it's written like 10 to 50 nanometer range. Now, how do we get this gradation? Uh, Under what circumstances we'll get a 10 nanometer particle, under what circumstances we'll get a 50 nanometer, or in some cases we can get above 50 as well. Now we'll see that in the next slide. The other method is brush Griffin method. In this there would be two-phase system: water organic, where gold salt is reduced by sodium borohydride. This one is a weak reducing agent in the presence of thiol ligands, producing small stable nanoparticles. Like you can follow this protocol if you want to synthesize gold nanoparticles of 1 to 5 nanometer range, like ultra small gold nanoparticles. The other method, before looking for the other one, let's first see what role does citrate play in this nanoparticle synthesis, and why it was written that in this case citrate act not only as a reducing agent but also as a stabilizing agent. So in the famous Turkevich method for making gold nanoparticles, citrate reduces Au3 plus to Au0. So yes, it is a mild reducing agent. At the same time, citrate ions adsorb on the nanoparticle surface and provide negative charge. You see this? And provide negative charge which stabilizes particle electrostatically and prevent agglomeration. So because there would be these particles adsorbed on the gold nanoparticle surface, there would be no agglomeration. So citrate has a dual role. It not only acts as a reducing agent but also act as a capping or stabilizing agent. Okay, so what role does thiols play? Like in the next method that we studied, in which we were using sodium borohydride, NaBH4, which is also again a mild reducing agent. And in addition to this NaBH4, they were in the presence of thiols. This reduction was happening. So thiols have a strong affinity for metal surfaces, especially gold and silver. They mainly act as capping agents that stabilize nanoparticles and allow functionalization, and they are not significant reducing agents. So in this case, thiol is acting as a capping agent because they have inherently strong affinity towards these gold and silver. Now, what is the why this capping and stabilization is needed? Now, capping stabilizing agents adsorb onto the nanoparticle surface and provide repulsive forces. Like in the previous slide also I explained you people. Like suppose these are your nanoparticles. Now, they tend to aggregate because of high surface energy. Now, but when there is a capping layer, what would happen is these repulsive forces, because of this capping, there would be significant repulsive forces, and because of the repulsive forces, these nanoparticles won't come together or agglomerate. So, keeping particles separated, there would be repulsive forces because of which the particles will stay separated and as well as stable in solution. So, it prevents nanoparticles from degrading, oxidizing, or growing larger during storage, improves colloidal stability. In addition to that, it also improves the colloidal stability, that over the time once you store it, or they would not, they would the nucleation would be stopped and they won't grow bigger in size or they won't agglomerate. Now, some capping agents like thiols, polymers, biomolecules offer functional groups that can be further used for bioconjugation, sensing, or drug delivery. Now, this one is an important point, and this will be studying. Suppose this is your gold nanoparticles, and whatever capping agent you have used to stabilize it, suppose it has some functional groups on it. Suppose that it could have OH, it could have, suppose if there is some protein or amino acid, there could be NH2. Now, these functional groups, presence of these functional groups open avenues for further biconjugation. Suppose you can utilize this particular functional groups and add some targeting moieties on it. Now, suppose you add some targeting moieties, and these targeting moieties are specific to targeting some of the, suppose suppose you have added some of the targeting moieties that target breast cancer cells, some of the antigens that are overexpressed, or some of the molecules that are overexpressed on, suppose this is a breast cancer cell and they have something overexpressed. In case of cancer, there are some different biomolecules or precursors that are overexpressed on these cancer cells. Now you have added some targeting moiety on this gold nanoparticle so that you can target these, breast cancer cells. So one thing that you need to keep in mind is sometimes these capping agents offer functional groups which can be used for further biconjugation. You can add something, you can tag something on these gold nanoparticles. Like we'll also study, like in later sections we'll study surface plasmon resonance, we'll study how this gold nanoparticles contribute in this LSPR. It has LSPR property, localized surface plasmon resonance, which is used for Raman spectrometry. Then surface plasmon resonance, which is a lot of times used in this the kind of kits that are being made for detection. So we'll also study that. Now, the next method we saw how the chemical based synthesis of gold nanoparticles. The other one is green synthesis. So, as we all know, in case of green synthesis, there are no chemical precursors involved. So, in green synthesis, plant extracts, microorganisms, or biomolecules act as reducing and capping agent, offering eco-friendly production. So, why is it called green synthesis? Because there is eco-friendly production. So, suppose these are plants, bacteria, fungi. So, suppose there is some plant. There are different plants whose it's not just whole plant. Sometimes root, stem, leaves. The extract of root, stem, leaves, etc. can be processed, and then you can add this HAUCl4, which is basically the source for gold. And instead of any reducing agent, like there's no involvement of any chemical reducing agent, and these can be reduced, and you can get your nanoparticles. So whenever we are using plant, bacteria, or microorganism, we label it as green synthesis or the biological means for synthesis of nanoparticles. Now, one important thing or one interesting thing I would say, just a second. Okay. So one interesting thing is, see, you would be seeing different colored, solution. These are nanoparticle suspension, and you see different sized nanoparticles. Now, all of these are gold nanoparticles only. The difference is the size is very small. When the size is very small, like suppose in the range of 10 nanometers or below, the color that you would see would be red wine in color. And as these nanoparticles grows in size, like if you are getting a larger nanoparticle above 50 nanometers, it would start turning purplish. So because see, it's in nanometer range, you cannot see it by naked eye. So obviously you have to perform the characterization, you have to do the electron microscopy to actually visualize of what size these nanoparticles are formed. But before that, as a preliminary means, suppose you are getting, suppose in this image, if you look, if you are getting this kind of color, even before you have not done electron microscopy to see the image of nanoparticles, even before if you have not checked its zeta potential value, its size and everything, its absorption spectra, just by looking at the color, just by looking at the color, okay, if you're getting this red wine of color, you can say that the particles could be somewhere in the range of 20 to 30 or 35 nanometer range. So, and how do we ensure such kind of size tunability? By varying, you know, the precursor concentration, like what concentration of HAUCl4 we are using, then reducing agent strength, if we are using citrate, if we are using sodium borohydride, and in what amount as well, how much reducing agent we are adding. If we'll add more of reducing agent, it will be further reduced. And then stabilizer, what capping agent we are using and in what amount. So all of these factors, all there is a crucial interplay of all these parameters, which through which you can easily tune the size of gold nanoparticles that you are getting. Now there are different colored gold nanoparticles. What does this show? Gold nanoparticles show different colors due to surface plasmon resonance. Small nanoparticles appear red, while increasing size or aggregation, whenever there is aggregation shifts surface plasmon resonance to longer wavelength, giving purple or blue colors. Now, what are the different applications of gold nanoparticles? So this one, it can act as a linker. Just now I explained, like if there are some capping agents or surfactants that are being added on the gold nanoparticle surface and it offers some functional groups. These can be used as, for linking or for biconjugation or for adding other groups. Like it binds thiol and amine groups, allowing attachment of drugs. There could be some drugs which can come and bind with those functional groups. There could be attachment of DNA, antibodies, or targeting ligands. Now, next is antigen binding. So, gold nanoparticles conjugated with antibodies are used in immunoassays for specific antigen detection. Like just now I told you, there are different rapid diagnostic tests. So, these are kit-based test, like there are, there would be gold nanoparticles with antibodies, and when you'll pass the sample, they would have antigens, they would come and react with it, and maybe by fluorescence or something, you can detect it. We'll study this as well in later sections, because this particular week, we just have to discuss how these different nanoparticles are synthesized, what are the different synthesis mechanisms, and these things. Now, drug carrier, as a drug carrier, how deliver anti-cancer or antimicrobial drugs with improved targeting and controlled release. It can, it can be used as a drug carrier. We can bind the drug on gold nanoparticles and help it deliver it. Gene delivery. Okay. Gene delivery, bind and protect DNA, RNA for cellular uptake in gene therapy. The other one is shape recognition. So different shapes, spheres, rods, stars interact differently with cells, enabling selective targeting and sensing. Now, one thing that I want to discuss is, maybe a lot of you people already know. It's not like whenever we're talking about nanoparticles or gold nanoparticles, these are spherical in shape. Now, gold can be synthesized, like gold, these nanoparticles can be synthesized in different shape. There are gold nanorods, there are gold stars. So there are different shapes of gold nanoparticles. And maybe when we'll discuss more about the surface plasmon resonance or LSPR property, you'd be better, you'll better understand what does these different shapes do. Yes, you'll be getting the recorded session on YouTube as well. So fluorescent imaging, strong surface plasmon resonance enhances optical signals useful in bioimaging and cellular tracking. Suppose, okay, now suppose understand, okay, maybe I think when I'll teach you the surface plasmon resonance, because it's something very interesting topic for you to understand what exactly is LSPR. Maybe you can read it on own as well, or I'll also be teaching LSPR and this SPR property, and how it can be useful. Now, try attempting this question, because in these questions are like kind of very basic. Try attempting it and write in the chat box. Try attempting the question. Okay. Right. So yes, the correct option is they have unique optical, electronic, and catalytic properties, because as we know, these are noble, like gold nanoparticles. They are not easy to degrade. Then also they are not cheaper, because we are talking about gold. And this is also wrong, like they do not interact with light, because just now we studied the surface plasmon resonance property. When we'll study about it, we'll see like it very effectively interacts with light. So the correct one is they have unique optical, electronic, and catalytic properties. Now try attempting this one. Just now in the previous slide, I have explained it, how different parameters, you know, affect the kind of gold nanoparticles that forms. Try attempting it. Good. If anyone has any doubt also about any of the topics that we have discussed like till now, you can also ask it. So yes, the correct option is both A and R true, and R is the correct explanation of A, because just now we have like kind of seen that the size of gold nanoparticles can be controlled by adjusting the reaction parameters. This is completely correct, because by adjusting the concentration of the precursor, the strength of the reducing agent, the capping agents, we can control the size of gold nanoparticles. And there are different parameters such as pH, then temperature, reducing agent concentration. All of these things influence the size and stability of gold nanoparticles. So yes, the correct answer is both A and R are true, and R is the correct explanation of A. This one. Moving forward, the next topic was biological synthesis of nanomaterials. Now, briefly, we have already seen that what does this mean? Briefly, we have already seen what does this mean, biological synthesis. So there are involvement of microorganisms like yeast, fungi, sometimes yeast extract are also being used, then bacteria and all those things for synthesis of nanoparticles, biological synthesis of nanoparticles. And in addition to that, like microorganisms, there are plant tissue, the extract of different plant tissues like leaf extract, stem, root, fruit, peel, flower, all these extracts are being used for synthesis of nanoparticles. So, um, antimicrobial, antipathogen, mosquitocidal uses. Then, in addition to that, some of these are also being used for cosmetics and medical appliances. Like sometimes we'll see the example as well, but for example, there is a branch, or I would say a research area, where people are kind of they want to replace, like suppose heart stents are there in case of atherosclerosis, or if there is collapse of some of the arteries or nerves or blood vessels basically. So we apply stent there. So instead of using the normally available non-biodegradable stents, people are kind of developing some of the nanomaterials, basically some of the biological materials which can be applied there, which can be used for synthesizing that, and since it would be biodegradable, so over the time it would be dissolved. So these are the approaches that are being used. Then for photoimaging, photothermal therapy. I hope some of you might be knowing about photothermal therapy, but those who do not know, photothermal therapy basically refers to, suppose if you see this mice, it has tumor here. Okay, it has tumor. It could be breast tumor, or suppose it has breast tumor. So what we do is we inject some nanoparticles, and these nanoparticles have a characteristic that when you will shine near infrared light on it, suppose you injected it, you can inject it at the tumor site also, you can do the injection via IV route also. And if your nanoparticles have this capability of particularly accumulating and targeting the tumor site, they would come and aggregate here. Or if they do not have the targeting ability, you can simply do the intratumoral injection, means directly into the mice tumor. And these nanoparticles have this characteristic that when you shine the near infrared light, like uh, above 700, 750 nanometers, like in the infrared range. So when you shine the near infrared light, which is basically done by using the NIR laser, because they have strong beam, when you shine the near infrared laser, what would happen is these nanoparticles convert this into heat energy. And because of hyperthermia, because of heat, this tumor would be like eradicated. So I would, because my thesis is based on this photothermal therapy, so I have done all these animal studies. So maybe in the next section, if you guys will come, I'll show my personal like the thesis data, because we have got excellent results for this one. I have done the like somebody wants to ask me. Okay. So I have particularly done this animal work in which we developed tumor in mice and we treated it through photothermal therapy. So maybe since because it's just an introductory session, introductory live session, so I'm just restricting myself to the course content that is designed for you guys. But since we are like once we'll move towards more of the application side, I will definitely show those data here in the like and also share what are the limitations of it, what difficulty we faced, and also I'll show how effectively the tumor was reduced, how we can get rid of the tumor, like the solid tumor that we developed in the mice. It was in the control groups. As the days progressed, the tumor like grew in size, but in the ones that we treated, there was very significant reduction in the tumor size. So I'll show that data now. Another is magnetically responsive drug delivery. Then various types of human cancer. So a lot of times we use these biological materials for synthesizing nanoparticles, which are used for different applications, like for disease treatment, for as anti-cancer agents. Then nanosensors detect biomolecules, environmental factors. So all these are applications of biological synthesized nanoparticles. Okay, so this biological synthesis of nanoparticles we have already discussed since it's an eco-friendly kind of approach. So that is why we label it as green synthesis. So it uses living organisms or biological extracts to reduce metal ions into nanoparticles in an eco-friendly manner. So plant extracts are widely used in green synthesis of nanoparticles because they naturally contain bioactive compounds. So the bioactive compounds they carry in itself, they act as reducing agent, which can reduce metal ions, and stabilize the formed nanoparticles. So this can be done by using microorganisms like bacteria, fungi, algae. They reduce metal ions either intracellularly or extracellularly through enzymes and proteins. So like Fusarium oxysporum is used for synthesis of silver or gold nanoparticles. Using plant extracts like plant leaves, fruits, roots, they contain phytochemicals like different alkaloids, flavonoids, phenols, terpenoids, which act as reducing agent and stabilizing agent. Means they act both as reducing and stabilizing agents. Then using biomolecules, like if you'll go for the academic, like the journals, the research papers that are currently being published, and those who are specifically highlighting the green synthesized nanoparticles, you would see a lot of protein based, amino acid based, or sugar based, like Kedson nanoparticles, PLL nanoparticles, BSA nanoparticles, HSA nanoparticles. So these are basically human serum albumin, bovine serum albumin. So all these are proteins which are being used for synthesis of nanoparticles. There are different routes for their synthesis as well. A lot of that, those approaches are also discussed in the pre-recorded sessions that are available in your NPTEL website. So as we discussed, these are eco-friendly. They offer low toxicity, mild reaction conditions, and they have, and most importantly, okay, this one is good biocompatibility. Because the thing is, when we talk about metallic nanoparticles, okay, metallic nanoparticles or any of the inorganic nanoparticles, if you're talking about metallic nanoparticles, it's fine. Like at some concentration, it would have good biocompatibility. It is not cytotoxic. But even if we talk about some of the mostly used nanoparticles, like suppose if you're talking about gold nanoparticles, there aren't enough studies that can prove or that has like kind of verified its long-term cytotoxicity in body. We don't know its long-term fate. Like suppose if these gold nanoparticles are being used in human body for some treatment or for something, so long-term cytotoxicity studies are not available. So we do not know that as of now. So in those instances, that is why the focus is more on this biologically synthesized nanoparticles or amino acid based or protein based, which are biocompatible, and in addition to being biocompatible, they are also biodegradable. These are biodegradable as well. So this needs to be taken into account. So here also the same mechanism is shown. Leaves, roots, we get the plant extract or powder. Then we use biological molecule stabilizing agents. We heat it, we add this precursor, and then we get silver nanoparticles. So now let us understand how plant extracts help in green synthesis of nanoparticles. We discussed also, but please do remember what are the different bioactive compounds or phytochemicals that are present in plant extract which help in synthesis of nanoparticles. So these are flavonoids, phenolic acids, terpenoids, alkaloids, tannins, proteins, and sugar. So these compounds, these these phytochemicals which are present in plant extracts, these compounds donate electrons and reduce metal ions, means it functions as a reducing agent. For example, in silver nanoparticle synthesis, phytochemicals reduce Ag plus into metallic silver. So these are the compounds that contribute as a reducing agent. Anyone wants to say anything? Just a second. Okay. So why do we use plant extracts and why these natural sources or are more preferred? Because they have natural capping and stabilizing agents. The same biomolecules bind to nanoparticle surface and as well as act as capping agent which prevents segregation. Then we can control particle size and shape. This eliminates the need of synthetic stabilizers because they act both as capping and stabilizing agents. So do not need further surfactants or further any different synthetic stabilizers. Eco-friendly and safe. Since they are plant-based, so there are no toxic chemicals like we saw in case of gold nanoparticle synthesis through chemical means. We were using sodium citrate or sodium borohydride. So in this plant-based synthesis, you do not need these toxic chemicals. It requires mild temperature and pressure, produces less hazardous waste. Then there are plants are abundant, renewable, easy to process, and suitable for large scale production as well. Try attempting this question. I would request everyone to kindly attempt doing it at least. It's not necessary you have to be right. The question reads as in a green synthesis experiment, a team uses fungal extracts to synthesize nanoparticles. Okay, so here they are using fungal extracts, which obviously might be containing some of the bioactives or some of the bioactives which can help in reducing which can act as reducing agent. So they observe that proteins in the extract stabilize the nanoparticles while smaller compounds like glucose and amino acids facilitate reduction. What mechanism likely stabilizes the nanoparticles? So they are saying that they observed that proteins kind of stabilize the nanoparticles and that too greater than 3 kilodalton size proteins. But smaller compounds like glucose and amino acids they facilitate reduction. What could be the possible reason for it? So, in order to correctly answer this question, we have to understand the mechanism. What role does this size thing plays in stabilizing the nanoparticles? So, the correct answer obviously would like we'll come to the correct answer after understanding what exactly the mechanism is. Just a second, I think. Okay, I think we have one more question related to it. That's why I haven't put the answer here. I'll straight away go first to that particular question in which we have that answer. Okay. So same question, same related question is this one. Which component of fungal extract acts as a capping agent to stabilize nanoparticles? So we just now saw this proteins 3 kilodalton. Now the question arises, why only proteins? Why not glucose? Why not amino acids? And why not sucrose? So let us understand the underlying reason, which is, in case of, like, there are three primary reasons. The first one is surface binding via functional groups, which is proteins. When we're talking about proteins, they have multiple functional groups: amino group, carboxyl group, thiol group, and this OH hydroxyl group. These groups, what does these functional groups do? They can chelate metal ions like Au3 plus and Ag plus and bind to nanoparticle surface, which forms a stable layer that prevents segregation. So as we have seen earlier also, like, these particular groups have special affinity towards this gold and silver. They bind to the surface and then kind of form a stable layer which prevents further aggregation. So one of the reasons is surface binding via functional groups, which are surfaced or which are provided by the proteins. But in case of glucose, sucrose, and amino acids, they have very fewer binding sites. They do not have more binding sites just like proteins. Sugars mainly have OH groups, weak adsorption, and single amino acids may have only one or two functional groups, which leads to insufficient stabilization. So because of the presence of the functional groups that are being surfaced by these protein molecules, they act as stabilizing agents. The other factor is steric stabilization. As you can see here, it's clearly mentioned greater than 3 kilodalton. So proteins are large macromolecules. Once adsorbed on the nanoparticle surface, they provide steric hindrance. Suppose this is your nanoparticle. Like, just assume, and then suppose there are small molecules, no proper binding, and all those things are there. So whenever there would be like nanoparticles, similar nanoparticles, there would be much less repulsive forces, and tend to they would tend to kind of aggregate. Now, in this case, proteins are large macromolecules. Once absorbed on the nanoparticle surface, they provide steric hindrance, preventing particles from coming too close and aggregating if the molecules are small. Now, suppose these are the nanoparticles, and now you have protein molecules. Like, we'll also study about some more. These are large macromolecules. Now, because of this, the steric hindrance would be less. Sorry, the steric stabilization would come into the picture, and they would not tend to come together or aggregate. So that is why proteins greater than 3 kilodalton act as good stabilizing agent. So preventing particles from coming too close or aggregating. Small molecules like glucose or amino acids are too tiny. These are too tiny to provide this steric barrier. So in this case, proteins, they kind of provide the steric barrier. The next thing is electrostatic stabilization. Many proteins carry charged residues at physiological pH, like suppose this is NH2, NH2 plus, CO minus. They have charged residues at physiological pH, okay, at 7.4, which create electrostatic repulsion between nanoparticles. Simple sugars have neutral charges, so there would be minimal electrostatic stabilization. So if there would be electrostatic repulsion, again, because of the electrostatic repulsive forces, they would stay away from each other, and these particles would not aggregate. So this is the mechanism, this is the reason why these proteins only, act as stabilizing agent. They stabilize the nanoparticles. So I think you all should remember that because they furnish functional groups, they ensure there is steric stabilization as well as electrostatic stabilization. Now coming back to where we left, we were discussing this question. Now coming to the synthesis of carbon dots. So what are carbon dots? Carbon dots are tiny carbon-based nanoparticles, typically smaller than 10 nanometers in diameter. So these are very small nanoparticles. Like all carbon dots, whenever we're talking about carbon dots, they are mostly less than 10 nanometers in diameter. They are best known for their strong and tunable fluorescence, meaning they glow under UV or visible light. So a lot of time these carbon dots are used. Suppose carbon dots or quantum dots, these are used for detection purposes also. Like suppose whenever these carbon dots come in contact with a particular analyte, suppose, so they would glow, they would give fluorescence, and because of which it can be used for some detection or for a lot of other applications. I just gave one example. So the key properties are these are fluorescent when they come in contact with, under specific conditions, they become fluorescent, which can be used for detection purposes. They are water soluble, low toxicity. Now again, low toxicity, but the dose you need to decide. Just a second, just give me 1 second. Just a second. Okay. So, because of these properties, carbon dots are widely used in bioimaging, drug delivery, sensors, photocatalysis, LEDs or optoelectronics, and environmental monitoring. So, okay, these are the basic applications where these carbon dots are used. How these are used, we'll study this. So, if we talk about the application of carbon quantum dots, so this figure basically summarizes the multifunctional biomedical uses of carbon quantum dots. They can be used for bioimaging. How they can be used for bioimaging? Just now we studied that we can fluorescent labeling of organelles like mitochondria, lysosomes, real-time cellular imaging due to strong photoluminescence. So these carbon dot labeling of mitochondria, we can label it with that, and once we have labeled it, at least we can see, we can see under microscope, we can do bioimaging, and this one would give fluorescence. Then therapeutic, like photothermal therapy, carbon dots can be used for photothermal therapy as well. Like when we'll shine light, it would produce heat, and because of the hyperthermia, it would kill the cancer cells. Drug delivery carriers, anti-cancer drugs like cisplatin. So we can kind of, attach the drugs or maybe load in the carbon dots, the anti-cancer drugs, which can ensure drug delivery. Then for sensing, like just now we discussed the metal ion detection. Like suppose there is heavy metal contamination in water bodies. So a lot of times these carbon dots or quantum dots are used for this heavy metal detection, like the contamination. If you have contaminated water and you want to see what are the different heavy metals uh, that has contaminated that particular water source, with the help of carbon dots we can do that. Once these carbon dots would interact with the heavy metal ions, they would give fluorescence. Then pH responsive fluorescence sensors. Other one is antibacterial activity. It can inhibit or kill bacteria via ROS generation. ROS means reactive oxygen species or membrane damage. The other one is combined theranostics. Like obviously, theranostics means it is performing the role of as a therapeutic as well and for imaging as well, like for diagnostic purposes. That is why therapy plus diagnostic, which means theranostics. Like we can do bioimaging as well, we can do therapy as well with carbon dots. So not only carbon dots, there are a lot of nanoparticles which are currently being strategized, which are currently being engineered in a way that they can be used as a theranostic agents. Like you can do therapy as well, you can also do diagnostic, like at what location those nanoparticles are going by means of maybe fluorescence imaging or Then there is this photoacoustic imaging thing also available. So a lot of things are there. It's very interesting. Now there are different types of carbon dots. This is graphene quantum dots. This one is carbon quantum dots. This is carbon nanodots, carbonized polymer dots. So these graphene fragments less than 10 nanometer, very high crystallinity, strong quantum confinement. Now this term, this quantum confinement, this is very important. In just next slide, we'll be studying this. What does this quantum confinement means? And clear lattice fringes. So basically what you have to understand is uh, there are different types of carbon dots, like which material is there, graphite quantum dots, carbon quantum dots, then carbon nanodots. They are mostly amorphous carbon and they mainly fluoresce from surface defects. Whatever surface defects are there, because of this surface defects, they make it unique, and because of this, it fluoresces. Low crystallinity. Then polymer carbon hybrid structure. Carbonized polymer dots. They are a hybrid kind of structures, contain molecular fluorophores and very low crystallinity. So these are the properties, but just remember the types, the different types of carbon dots. Now let us understand what does this quantum confinement effect means. So suppose whenever material size reduces to the nanometer scale, suppose this is the bulk material, and over the time, if the size is reduced to the nanometer scale, this one, typically less than 100 nanometer, electrons and holes become confined in a very small volume. In bulk material, energy bands are continuous. So what happens is, in a bulk material, energy bands are continuous and valence band to conduction band. But in case of nanoparticles, as you can see in the bulk material, this valence band and conduction band, there are continuous bands. Okay, energy bands are continuous. But in case of nanoparticles, the reduction in size leads to discrete energy levels. There are discrete energy levels, as you can see, rather than continuous bands. So what happens on the band structure? There is band gap widening. As particle size decreases, as the particle size decreases, the conduction band and valence band separate more. You can see the conduction band and the valence band, they kind of separate more. So smaller particles exhibit stronger confinement and higher band gap energy. This explains why quantum dots of like cadmium, selenium, for example, emit different colors depending on their size. So this thing also regulates this phenomena that different sized quantum dots give, even if those quantum dots are made of same material. But if they differ in size, they would emit different colors. So this phenomena is known as quantum confinement. And what does this electron confinement mean? In nanomaterials, electrons cannot move freely as in bulk. Their motion is confined in 1D, 2D, or 3D depending on the nanostructure. This confinement alters electrical, optical, and magnetic force. So these are like different things, but for now you just have to understand what does this quantum confinement means, and this high band gap energy, the conduction and valence bands are not continuous. Now, uh, this particular figure, it shows that from CPDs to this graphene quantum dots, as we move from CPDs to GQDs, the degree of carbonization increases. And like this diagram shows the carbon dots family arranged by x-axis, increasing degree of carbonization, increasing surface functional groups or polymer character. Orders from low, high carbonization, CPDs, CNDs, CQDs, GQDs. Mostly polymer-like, lowest carbonization, amorphous carbon, more crystalline carbon core, highly graphitic, highly carbonization. So as you move right, as we move from CPD to CND to CQD or GQD, what do we observe? The structure becomes more graphitic and ordered, as I have shown in previous slide as well, that it becomes more graphitic, the degree of carbonization increases. But as you move up, like from CPD to GQD, surface functional groups and polymer features increases. So obviously there are more easy to, surface modification. So जैसे जैसे these surface functional group increase, more chemical groups like OH, COH, NH2 are attached to the surface. So once these functional groups increases, So once these functional group increases, what happens? Because once the surface groups will increase, surface functional groups will increase, what would happen? There would be higher water solubility, better biocompatibility, and since these functional groups will offer sites for further bioconjugation, they will offer sites for drug loading or sensing because they'll have functional groups, maybe NH2, OH, etc., which can offer sites for drug conjugation and fluorescence mainly from surface states. So polymer features increase means the structure contains more polymeric chains or cross-linked molecular networks. It signifies lower graphitic crystallinity, more molecular type emission, strong excitation dependent fluorescence, behavior close to polymer dots. So some of the important things that you can remember from this particular slide is like as we move from like low to high, this thing increases, and as the surface functional groups like the increase, what happens is it becomes more capable of further conjugating to for like to be used for drug loading or all those purposes. Try attempting this question. The question reads, how does acidic pH affect the synthesis of series? Carbon dots. I'll give 30 seconds. Okay. Okay, so let's discuss what's the correct answer. So the correct answer is it produces smaller, well dispersed carbon dots. So why does acidic pH, how does acidic pH affect the synthesis of carbon dots? It produces smaller, well dispersed carbon dots. So why? What is the reason? Why does it happen? So what happens in case of acidic pH? It protonates surface groups. So suppose if it has amine group, so it would become NH2 plus. So there would be protonation. Okay. So NH3 plus, sorry. So acidic pH protonates surface uh, groups, increases electrostatic repulsion, limits particle growth. Means just we see in case of nucleation. So suppose if this is the particle growth, there won't be further there won't be any further nucleation. This would not happen. So it limits particle growth. It limits particle growth at this place, producing smaller carbon dots. So proton at acidic pH, COH and OH groups on precursors are protonated, reducing aggregation. Slower nucleation and growth. Protonation limits particle growth. More nuclei form, but each grow less, smaller in size. Means at this size, more nuclei are formed, but they do not grow much. Like they do not, the nucleation is less because of which they do not grow in size, and smaller sized carbon dots are formed. Surface charge repulsion. So positive surface charge from NH3 plus groups enhances electrostatic repulsion and prevents aggregation. So this prevents aggregation and it also ensures smaller sized carbon dots are formed. So the overall effect that we see is more nucleation size but inhibited growth, which leads to formation of smaller uniform carbon dots. So this is the reason. The correct answer was acidic pH causes protonation of these surface groups, which in turn increases the repulsive forces, which in turn ensures smaller carbon dots, slower nucleation and growth, and no aggregation. Now, moving forward, try attempting this question. The question reads, during a carbon dots synthesis experiment, a team adjusts the pH to acidic level and uses PEG as a stabilizer. This is polyethylene glycol. It is one of the most commonly used capping agent and a stabilizing agent. The resulting carbon dots are small, well dispersed, and show strong fluorescence. What role does PEG play in this experiment? I think I already gave the answer, but I want you guys to answer it. Okay. Okay, so I guess most of you have given the correct answer. Good. So it prevents agglomeration and improves dispersion of carbon dots. So how does this happen? Now I have also attached a picture here, like this silver nanoparticles that are formed, and then there is these silver nanoparticles are being capped by PEG, that is polyethylene glycol. So just now we have also seen like there would be steric stabilization as well because of it. So the two particles, because of this uh, steric factors, like they would be highly, so because of this two particles there would be enough repulsive forces, they won't come together and won't aggregate. So it happens because we have added PEG as a stabilizer or as a capping agent. So long flexible polymer chains, as you can see, long flexible polymer chains surround the particle surface, which prevents aggregation by physical barrier. So there is, steric barriers are there because of these long polymer chains of PEG. Then it forms hydrated layer around nanoparticles. So since it forms hydrated layer, it also improves the colloidal stability of these nanoparticles in water. Then reduce protein adsorption. It resists binding of blood proteins, opsonization. Now, most of the time, what happens, there is this corona effect, there is this opsonization. So there is deposition of, blood proteins on these nanoparticles, which can alter its inherent characteristics. But by capping, by stabilizing the nanoparticles using PEG, we can prolong circulation time in vivo. Then chemical versatility, functionalizable ends. It allows conjugation with drug ligands or antibodies. And then biocompatibility, inert non-toxic polymers safe for biomedical application. Okay, so I think this one I have discussed. Okay, so the last topic was polymeric nanoparticles. Polymeric nanoparticles. So polymeric nanoparticles are extremely small particles. As the term denotes, there are long chain of polymers and we kind of trigger them or initiate them. So these polymer chains kind of get folded and form the nanoparticles like this. So they can form a matrix, and in this space we can load the drug of interest, or in some cases we can add the surface also, because when this happens, the the hydrophilic core comes inside, the hydrophobic core gets inside the hydrophobic groups. So in this you can also add drug moieties at the surface, or it can also be used as a cargo. We can load drug inside as well. So it ranges, the size ranges from 10 to 1000 nanometers, made from natural or synthetic polymers. Because of their tiny size and customizable surface properties, because we can regulate these, we can do the surface modification. They are widely used in medicine, drug delivery, biotechnology, and material science. So they are composed of biodegradable or biocompatible polymers. Has someone written? Okay. These, they are composed of biodegradable and biocompatible polymers. So it's not limited to this only. There are many other polymers also which are used, like PLA, PGA, PLGA, chitosan, alginate, natural poly. These are the natural polymers. So these polymers are used for synthesis. So there are two types. One is this nano capsule that I explained. This is nanosphere. So polymer nanoparticles are formed. So in this case, this one is labeled as nanocapsule, in which we are putting drug inside, and then there is a polymer membrane around it. And then this is a nanosphere in which there's polymer matrix, and then drug is being loaded here. So solid matrix system, drug is dispersed throughout the particle. And in nanocapsule, sorry, in this one, and in nanocapsule, pore shell structure, drug is confined in a central cavity surrounded by a polymer membrane. So now there are different methods of forming polymeric nanoparticles. There are different synthetic synthesis mechanism as well. So suppose here they have formed PLGA PEG in acetonitrile. Now we know PLGA is a polymer that is used for synthesizing polymeric nanoparticles, and PEG here functions as a stabilizing agent. Now see, this kind of nanoparticles are formed. This blue thing signifies PLGA, and this red linkers, these are PEG. So which are on outside. So maybe it would ensure more of the repulsive forces. There would be no aggregation because of the presence of this red colored PEG. So there are different methods. Like in this case, they have made this via rotary evaporation. So this polymer is dissolved in an organic solvent. Like in this case, here it is acetonitrile, emulsified in water. Solvent removed by rotary evaporation. Then the solvent is removed, and what is left behind? The nanoparticles. Solvent evaporation method hai ye. This one is solvent evaporation method. It causes controlled polymer self-assembly into nanoparticles. So how does this happen? First of all, an organic phase is prepared. Like we dissolve PLGA PEG in volatile organic solvent. Do remember volatile organic solvent, because we are following the rotary evaporation method. Then if you want to, in addition to this, if you want to add drug, so you can add drug, prepare aqueous phase, water containing stabilizer. Then we have to do emulsification. This one is an important step. Add organic phase into aqueous phase. So drop by drop, suppose this is your aqueous phase. इसमें in this we'll add the organic phase drop by drop, sonicate or homogenize. Suppose this is kept on a sonicator, or like this thing needs to be sonicated or homogenized, which forms oil water emulsion. Transfer emulsion to rota vapor. Then this emulsion that is prepared after adding organic phase to aqueous phase, this is transferred to this rota vapor. This is the, you know, rota vapor, which is then connected to the machine. So what does this rota vapor do? This evaporates organic solvent under reduced pressure. And as this solvent evaporates, polymer precipitates and nanoparticles are formed. Okay, these nanoparticles are formed. So then once these nanoparticles are formed, they are centrifuged and washed to remove excess stabilizer, and then stored. The other method is by use of ultrasonic atomizer. If you remember, when I was telling about like reproducible or monodispersed nanoparticles with narrow size range. So this ultrasonic atomizer, so this is the syringe pump or controller. We regulate the flow, like at what rate the uh, liquid shall flow from it, and then this is the magnetic mixer or magnetic stirrer with a magnetic bead in it that continuously rotates or produces agitation. So it produces very fine droplets using high frequency. We set the frequency here in this, and frequency sound waves 20 kilohertz to 2 megahertz. These micro droplets act as tiny reaction vessels where nanoparticles form after solvent evaporation or cross-linking. This technique is commonly used for preparing polymeric nanoparticles and microspheres in a controlled and scalable way. So polymeric nanoparticles, why these polymeric nanoparticles are especially valuable when we talk about drug delivery applications or this biomedical field? Because whatever nanoparticles are formed, like the drug inside these nanoparticles are safe, and it can be effectively delivered at the target location. It improves drug solubility, control and sustain release, because if you are giving drug as it is by oral means and all those things, the bioavailability at that target location is very low. The bioavailability is very low. So that is why this nanoparticle based approach is followed these days for these drug delivery application. Control and sustain drug release over time. Like once you have loaded your drug in your nanoparticle. So what would happen? It would reach the target site and it would slowly release the drug at the target site. So there would be sustained release of drug at the target site, which would obviously improve the outcome. Then target specific tissues like tumors. So suppose you have this nanoparticle, you loaded some anti-cancer drug in it, and then you added some of the precursor molecule or receptors, and suppose these receptors with which you have surface functionalized these nanoparticles, you have attached these receptors on the surface of these nanoparticles, they have tendency to go and specifically target cancer cells. Suppose some of the molecules which are overexpressed on these cancer cells, these target molecules go and bind here. Now, in this case, there is target specific. We are synthesizing nanoparticles that targets a specific location, and once it has reached to it this specific location and binded here, it will release drug and it will kill the cancer cells. More often, it has reduced side effects, obviously because it's not immediately going to the bloodstream. Now, there are different methods for this preparation as well. Like one is solvent evaporation method that we studied, like dissolve polymer in volatile organic solvent, add drug, then ultrasonic atomization. Feed solution to ultrasonic atomizer, fine droplets are sprayed. So the droplets are sprayed here. You supply the solution here, it flows at a particular speed, you set the frequency and everything, and then these are sprayed as a spray. This is a collection media. So fine droplets are sprayed into a chamber containing air or aqueous stabilizer solution. Organic solvent evaporates rapidly and polymer precipitates and hardens into nanoparticles, which is then followed by centrifugation and washing. So this method, solvent evaporation, is used for poorly water-soluble drugs. The other one is ionic gelation or cross-linking method. This is for hydrophilic polymers like chitosan. So in this, chitosan is dissolved in dilute acetic acid like TPP, and then again this we spray polymer solution into cross-linker solution. Electrostatic interaction forms nanoparticles immediately. So here there is precipitation happening, and then there is electrostatic interaction. This is basically done for protein, DNA, and vaccine delivery. As you can see, this is ultrasonic atomizer. So it generates ultrasonic waves which, through which the solution is passed and sprayed. This spray collected in a cross linker kept on magnetic stirrer. So this is a magnetic stirrer. In this particular vessel, there is a cross linker solution kept. So as these small aerosol-like small particles form on these cross linkers, these nanoparticles form via this interaction. Now, suppose this is like from the graphical abstract of one of the papers. So they have kind of studied the drug release. Like they have loaded this piperin drug in PLGA nanoparticles. Now, one important thing is once you have loaded your drug in nanoparticle, you also have to check if this drug is being released at a target site as well. So for this, we do the drug release study. So you analyze like after 50 hours how much drug is released, after 100 hours how much drug is released, after 200 hours how much drug is released, because we are talking about continued and sustained drug release at a particular location. So these studies are also important. Then the next thing is inherent biological properties. Now, what these polymeric nanoparticles are capable of? So they have mucoadhesivity. Some polymers interact with negatively charged mucal surfaces, prolonged drug resistance in time. If there are some polymers, suppose these are polymers, you form nanoparticles with it, and now since they are mucoadhesive, means wherever there is mucus, they would go and there would be proper adhesion with it. So suppose you want to make you want to do oral delivery of drugs. So as we know that the internal linings of our mouth, they have mucus. So if we want to do oral delivery, we can make a patch of these polymeric materials and which would be mucoadhesive, or for ocular purposes for intestinal mucosa, like wherever there is mucosa and we want uh, you know any polymer or nanomaterials to be used there for drug delivery applications, we can synthesize them with this mucoadhesive polymers. Then hemocompatibility. Hemocompatibility means whatever nanoparticles or nanomaterials that you're synthesizing, they are hemocompatible, means they do not cause damage to your red blood cells. So which would make it safe for intravenous administration. Then it should be non-toxic and biodegradable. Means there should be no toxicity and it should be biodegradable. That over the time it should itself undergo biodegradation so that it does not cause long-term cytotoxicity in body. Then some of the polymeric nanoparticles have this antimicrobial activity as well, as they rupture the bacterial membranes. And because of this property, this makes it useful in wound healing, antimicrobial coatings, and reduce infection risk. So mucoadhesive polymer, just like I explained, like you can simply take this polymeric patch with maybe it could have drug or something, and you can put it in the mucosal lining like buccal, sublingual, lingual, and over the time it would keep on releasing drug. So these are some mucoadhesive polymers like chitosan, sodium alginate. Why they are mucoadhesive? Because they have these functional groups. Then biodegradable, these polymers are biodegradable. So they over the time they get degraded, like they can be used for dental implants, then the cardiovascular system, the stents and everything that I taught, then systems for controlled drug delivery, orthopedics for like as a patch or something that can be used, these polymers, as over the time it will like, you know, there would be no need of invasive surgeries to take these things out. Now they can over the time, after joining, they can dissolve, they can like they are biodegradable. Hemocompatible polymers, I just now explained, like whatever nanoparticles or nanomaterials you have synthesized, it should be hemocompatible, means it should not cause hemolysis. So these are the healthy red blood cells. If your nanoparticles are hemocompatible, your blood cell would remain healthy. But in presence of non-hemocompatible samples, suppose if your nanoparticles are toxic or not hemocompatible, it would cause lysis of your red blood cells. There would be hemolysis. So whatever nanoparticles, whatever nanomaterials you are synthesizing, you should, and if it has to be used for human applications, then you have to ensure that these are hemocompatible. It should not cause red blood cell lysis. Then antimicrobial polymers inhibit or kill microorganisms like chitosan, PHMB. These are some of the antimicrobial polymers that disrupt microbial membranes, alter permeability, or interfere with microbial metabolism. Now we are kind of approaching the end. Try attempting this question. So it reads, a researcher is synthesizing BSA nanoparticles using the desolvation method. They observed that the nanoparticles formed are homogeneous and stable in water. Ethanol is added during the process, followed by a low concentration of glutaraldehyde. What is the role of glutaraldehyde in this process? So since we have less time, let's discuss it. So the role of glutaraldehyde here is to cross-link the protein molecules. Now, how does this happen? So this desolvation means removal of solvent molecules surrounding a solute, like a protein or polymer, okay, which reduces its solubility and causes it to precipitate or aggregate. Because once we have removed this solvent, what would happen? There would be precipitation and nanoparticle formation. In simple terms, when a dissolvating agent like ethanol is added, it removes the water layer around the protein. Suppose this is aqueous solution of albumin, aqueous solution of protein, and when we start adding ethanol in it drop by drop, what would happen is it would remove the water layer around protein, and the protein becomes less soluble, because obviously if the water layer is removed, it will become less soluble. These will come together and nanoparticles would form. So there would be formation of albumin coservates, and then we'll add the cross-linking agent, that is glutaraldehyde, which will form stabilized albumin nanoparticles. So BSA is dissolved in distilled water, ethanol is added, and then glutaraldehyde is used as a cross-linking agent for formation of stabilized nanoparticles, and then they are purified by centrifugation and drying. Next question is, in an attempt To enhance drug stability, scientists encapsulate niclosamide. This is basically anti-helminthic drug which is used for tapeworm infection. And there are albumin nanoparticles in which this particular drug is encapsulated. Now they are asking, they observe a slight spectral shift compared to the free drug. What might this spectral shift indicate in terms of chemical interaction? Now, one thing you need to remember, whenever we record, suppose we are recording absorption spectra of a free drug and it somewhere comes around, uh, 400 nanometer, like suppose. But once this free drug, like suppose you are recording spectra of this nanoparticle carrying drug, there would be some sort of chemical interaction, there would be some sort of aggregation because of which this spectra could change. There could be spectral shift, there could be slight aggregation, and this spectral change basically denotes that your nanoparticles are kind of like preliminary means of checking that the nanoparticles are formed, and to differentiate between free drug and nanoparticles with drug. So the correct option for this would be formation of protein drug nanoparticle complex due to chemical cross-linking. Why does it happen? Like suppose I have only added a reference spectra here, this particular drug is not there, but if you'll see, like this one, this black thing is free dogs. This is a free drug. Now, when you have encapsulated uh, this free drug, like there are different nanoparticles. As you start adding, like suppose you see this purple one, okay? If you compare the black and purple one, there is some kind of aggregation happening. Same for this as well. Like this is dogs, okay? Then red one denotes ICA plus dogs, and then for the nanoparticle, which is purple one, if you compare this red and green with the purple one, there is some spectral shift that you would notice. So these kind of spectral shift happens because of the formation of this drug nanoparticle complex due to chemical cross-linking. So now I have given some sample questions as well, which you can try attempting, because I will share this PPT also, but we can also like quickly discuss, like this is a important formula which says that critical speed, it depends on mill diameter, not ball density or powder size. So the critical speed of a ball mill depends primarily on mill diameter. Then what would happen if we increase milling time in mechanical alloying usually leads to? So if this happens, there would be formation of nanocrystalline structure, because if we'll extend the milling time, it will also increase strain and refines grain and raises hardness as well. Then quantum dots are typically made of this, we have already discussed in previous slides, that they are made of semiconductor materials like cadmium, selenium, cadmium, sulfur, zinc. Then when the size of quantum dot decreases, its emission color generally shift towards blue. Why does this happen? Or as we have also studied this quantum confinement effect and everything, because smaller quantum dots have larger band gap. We have studied the quantum confinement, they have discrete energy levels. So smaller quantum dots have larger energy gaps and higher energy emission. So whenever there will be higher energy emission, there would be blue shift, because higher energy means short wavelength. We have studied like red have largest wavelength. So higher energy means short wavelength, and short wavelength means shifts towards blue or violet region. So the correct option is shift towards blue. So these are some of the very basic application based questions that you can attempt, that you can understand and answer only when the basics are very clear to you. So thank you. This is it for today's session. And if you guys have any doubts, we can discuss it now. If you guys have any doubts, you can write in the chat box, or maybe because this one is very basic, but as these sessions will progress with the following sessions, we'll move forward with rest of the sessions. This will get interesting. And one thing I would ensure that I would also put some of the like real-time kind of data that I'm doing for, because I'm also working in nanotech only. So I would put some of the data so that it becomes more of like you are seeing what exactly is being done in science and not just what's written in textbooks and all these slides. So, okay, so the next session, like there would be three different live sessions taken by three different like teaching assistants. So one session is today, like Friday 6 to 8. Tomorrow also somebody else would be taking the session, and I think on Monday also. But for me, uh, yes, there would be a session tomorrow, but it will be taken by someone else for the same topics only. So, like you can attend that as well. My sessions are will be on Friday, 6 to 8 p.m. Like for the next 4 weeks, and maybe one extra session where I'll try to give a summary, and more importantly, I would try to focus like what are the budding areas or budding aspects of nanotech, and what are the limitations, or most importantly, what are the obstacles that that is particularly preventing the successful clinical translation of all these nanomaterials, and why these nanomaterials are still not in market as much as we expect them to be, and what are the areas that needs to be addressed and all those things. So that would be interesting as well. So that's it for today's session. Thank you very much. Thank you.